If you look behind you here at this field out here, you see those yellow plants out there? I have some examples here. These were pulled from another field. Okay, and I just want to give you kind of progression here. Here are your plants that are gone. Okay, these are completely gone. And before they looked like that, almost certainly looked like this. You had your yellowed leaves going up the stem, and then you have green on the very top. The next stage here, you have these here, and you can see that you have some yellowing on the very bottom. Okay, and the upper part is still doing fine. Now, if you look at the roots on these, you're going to notice problems on the roots right away. And I'll pass these around. These are the ones that are the least badly affected. But you'll notice right away you've got a root rot on here. This is almost certainly Fusarium. It's got a reddish color, but we haven't done the lab work, so I can't tell you that with total confidence.
So that's what this is out here then?
I haven't looked at these, but I would assume it's the same thing because we have a lot of Fusarium root rot problems out here. And now the severe ones, again, you're going to see the exact same. You're going to see the exact same thing. Once they're totally dead, it's really hard to tell. But on this one right here, if you open this up, you're going to see a bit of a reddish discoloration, and very obvious reddish discoloration is highly characteristic of Fusarium. This problem has been difficult. I'll let Kevin talk about the resistance end of it, but in terms of host resistance. But in terms of fungicide efficacy for seed treatments, we continue testing seed treatments, but up to this date we haven't found something that has given satisfactory results. Not to say that you don't get some suppression. It's just if you're looking for something that really gives you a good satisfactory control, we haven't found that. And towards that end, what we have looked to do in this, to address this question is, I talked to Blaine here this last winter, I talked to Julie. I'm looking to establish a long-term crop rotation experiment to look at what type of crop rotation we need to manage an existing problem and to prevent this problem from building up. Okay, and towards that end, we generated, Julie dug up through her culture collection that she inherited from Rubella Goswami, and she dug up all the Fusarium cultures that she inherited from there, and we chose about 12 isolates from each of the major Fusarium species that are associated with Fusarium root rot here in Canada. Okay, but these isolates are all North Dakota isolates in this case. Okay, we grew them up on grain. Michael Schaefer's help and Billy, who works with me, we grew up 150 of these aluminum pans worth of grain. What we have done is we've established a split plot design out there, and one of them's inoculated, one of them's not. And the non-inoculated treatment, we have about, the first stand count we did at 5 to 6 nodes, we had 296,000 plants per acre. And the non-inoculated, we seeded at 330,000 pure live seeds. Okay, so you had 10% reduction from your pure live seeds, which is not too bad. We're really close to that 300,000 plants an acre. Okay, the inoculated treatment, we were down at 270,000 plants an acre at 5 to 6 nodes. When we assessed the stand again at 12 to 13 nodes, the non-inoculated treatment was almost exactly the same at 295,000 plants an acre. Our inoculated treatment had dropped to 265 or so thousand plants an acre. So the inoculation definitely took, at least in terms of the plant population, a difference. Now the difference wasn't as strong as I would have liked. We are going to be taking the root rot ratings here this coming week. Would have liked to have done it earlier, but it was so dry here, it was so hard to get in the soil. We now finally got some rain, so now we'll get in. We have 4 passes for each plot, okay? The 2 middle passes get harvested for yield. The 2 outer passes get assessed for root rot. And so we're able to go in there and take root rot assessments without having any impact on our yield. That's something we're starting this year. If we're satisfied with these results, we'll look to establish a parallel trial in Williston, okay, for a second location in the pea growing region. We'll see how this year's trial, how this trial out here went this year and decide on the basis of that how we proceed. With that,
well, what kind of rotation would work? Everything seems to be susceptible.
Well, so the question is what type of rotation would work. Everything seems to be susceptible. And so reviewing the research that has been done in Canada, it does suggest that rotating still manages this problem. The limited work that has been done suggests that it may very well be possible that you could have enough diversity in those Fusarium species that there may be some host specialization that we're not aware of. But at any rate, the limited work that has been done gives a hint that rotating it, rotating your crops does
with what?
Isn't effective. In this case, we're looking at rotating with cool season crops that would normally be rotated with these. I know these are all typically susceptible. I'm open to suggestions and what you'd like to see. This is the time where you can tell us what you'd like to see evaluated.
Well, the problem is farmers don't like cereal crops anymore.
Sure. Well, we can. I don't know how much dryland corn can be grown up by Williston and Scobie.
Yeah, there, but I'm sure.
So, but I'm happy to have rotation with corn and soybeans and such in there. Absolutely happy to have that. We have the room at this time to design that. At this point, what we did was we established a uniform footprint of field peas over the site, inoculated versus non-inoculated splits, and now this winter we'll get to decide what we do for rotations. You know, one of the major questions is how many years you have to rotate out of peas, but the other question is, well, what's the impact of different types of crops in that rotation? On my end, I'll continue testing seed treatments. From what I've seen up till now, I'm not convinced that's the answer. So, yes?
Does the Fusarium from wheat and corn cross over, or is it different?
So they're the same species. Corn is a somewhat different mix, but wheat and canola, exact same species. Okay, now whether or not, but the limited work that has been done in Canada has shown that at least for a few individuals, so these fungal individuals, and if fungal individuals is highly aggressive from a given species against peas, can oftentimes be not aggressive at all against lentils. And a different individual that's highly aggressive against lentils will not be aggressive against peas, even though it's the same species of Fusarium. Now the work that has been done is very limited. I would call the conclusions tentative at best, but if you have that type of host specialization, it opens the window for crop rotations to work. And then it's the question of how many years you have to rotate out of the peas, what are the crops you should be targeting, in what sequence. So.
And also, Michael, so this is a soil-borne disease.
That's right.
How long? What was the last susceptible crop out here to make this look like this?
Well, I think in this case, Blaine can tell us the rotation. I think
basically these fields have been largely peas, wheat, or something very similar to that. It's been a very tight rotation.
Yeah, honestly, this type of crop rotation work has never been done. It needs to be done, in my opinion. And so at this point in time, it's a self-funded study this year. We'll look for some funding if we decide the results are promising enough to go forward. Okay, so with that, I'll turn it over to Julie.
Well, as Michael said, I think he indicated that I helped him, and I sort of helped him, but my graduate student here, Jennifer, raise your hand. She's working on Fusarium root rot in peas, and she actually did all the legwork on this and got the isolates going and got them cleaned up and got them started for Michael and did the legwork, and Michael and I kind of coordinated that together. I will say, as Michael indicated, we got these out of the storage from my predecessor, and we really had no idea which of these were still pathogenic, how aggressive they were. And so one thing that Jennifer started doing basically to try to aid Michael in his ongoing crop rotation study is to determine which of those isolates were pathogenic on peas and which of those isolates of the 8 species, and like he said, approximately 12 isolates of each, were most aggressive. And interestingly, as Michael alluded to, there wasn't a lot of difference between species, but there in some cases was a large difference within species across isolates. And so this goes back to know your enemy. And so with this project, we hope to, anyways, depending as Michael said, how this progresses, we hope to be able to isolate from the peas or rotation crops going forward to determine not only the level of root rot and how that affects the Fusarium composition, but also what's there. We know what we put in, but is wheat pea rotation affecting what we get out versus whatever, like Michael said, what he comes up with as far as what an important rotation would be. So those species that we get out will really maybe tell a story. Hopefully, we hope that it will tell a story of, yeah, you're still getting root rot on the rotation crop, but it's not, and those species may affect peas, but are they really the most important ones? And so we hope to learn a little bit more about it's a complex. My predecessor did a survey across North Dakota and found that 8 or 10 isolate or species. Now, which ones are most important? And I think just because it was the ones that were found most commonly, depending on sampling time, that will affect what you get out, and there's a lot of factors going into that. So going forward, we will work together with Michael. We hope to design some molecular techniques so that we can easily identify species coming out of roots. We don't have to plate them. We don't have to try to identify them visually, which is an extremely different process. Jennifer and I spent a week in Kansas with international experts at a workshop learning how to identify these species. It's not something we want to do in a field this size. So we're hoping to develop some easier methods to make that happen and going forward to really enrich this project that Michael started. The other thing that's kind of Jennifer's side project, and I got her sidetracked on it, but she was successful. She got 3rd place in a postering competition at our North Central Plant Path meetings, and she's only been with us for about 8 months, so we're quite impressed with that work and some of this work she's done in conjunction with Michael. But her real project is to, again, as we've talked about today, know your enemy, and she's working in conjunction with Kevin, and we will try to identify, again, screening methods in the greenhouse. And we really stress the importance of the fact that these methods that we're using to screen in the greenhouse really need to be replicated in the field, and we need to have that correlation. It does us no good to screen in the greenhouse if we're not doing it in the proper manner. So we have started with 2 species, Fusarium avinaceum and Fusarium solani, which have traditionally kind of been what we feel is important. Of course, we're looking farther into that. And we will look at different screening methods and how the relationship is with the screening methods themselves, but then across species and across isolates. And then use some of Kevin's lines going forward to identify also some resistance to some of the species that really haven't been evaluated as far as breeding programs are concerned. Correct me if I'm wrong, but we as a research community have identified resistance to Fusarium solani, but now that we know that this whole other complex is out there, we have no idea if the resistance are the same. Initial observations would be that they're probably not. The genetic resistance is probably not the same to either of these pathogens, and so we will really are trying to kind of get at that, and so that, yes, screening in the field is ultimately the most important thing, but we have to have some sort of greenhouse mechanism in order to have some of these things take place in the off-season, in the wintertime, and in a more rapid fashion. And so we can screen. Right now, it's 10 days to grow inoculum and 10 days to grow pea plants in a little cup, and we can have an answer versus this. Now, obviously, that's a first step, but an important first step, and we understand the importance of that being correlated to what we find out here in field trial.